Yeah, it's a great pleasure to be here. Thank you, Max, and thank you, Uri, for inviting me. I'm very honored to be in this fantastic company. We have worked on DNA repair and cell signaling, and therefore, of course, also aging. And it's so when healthy and sick cells are damaged or inflamed by radiation, you see protein oscillate up and down. In particular, p53 and kappa B. I concentrate on p53 today. Why? High p53 prevents cancer but induces aging. Low p53 induces cancer and prevents aging. Very delicate balance. I come back to that. They oscillate because they are regulated by negative feedback loops. And what we have worked on the last 2 years is that condensate and droplets formed around damaged sites and are part of repairing. Because if you damage and you see the oscillations I have been thinking about for years, it has to do with repair. And when condensate form around damaged site, one of the condensate or droplet, I use the same words, might tend to outcompete others and grow. So only one condensate will grow and the other will shrink. It's called Oswald ripening. But due to the oscillation, we can prevent this Oswald ripening, and therefore we get optimal damage repair. And actually, we have fantastic experiment both at Taiwan and at Harvard, showing that this is the case. Here I show you gamma radiation of cancer cells from fantastic collaboration with at Harvard. Really, Uri was a pioneer to study these oscillations, actually educated Galit Behar. And you see these oscillations, single cells, they can always oscillate for a week. And, but at some point they stop oscillating, and that we think either the damage is repaired or they go apoptotic or senescence. So it has a lot to do with aging. Of course, the p53 is regulated by a negative feedback loop. It's just a little cartoon of the mathematics behind. A negative feedback loop have an odd number of negative regulators. There was a famous paper 20 years ago called Repressolator, built a negative feedback loop into bacteria. And we have a negative feedback loop in p53 because p53 induces MDM2 and they bind in a complex, so p53 cannot be a transcription factor. That's why it's negative feedback loop. Of course, p53 is a cancer gene. It's mutating in nearly all cancers, and it's just said that high p53 prevent cancer but induces aging. And it controls many genes. NF-kappa B is related to diabetes and inflammation, also oscillate after damage. And we have worked on doing the so-called genetic networks of these. Here's the p53, here's NF-kappa 2. And with my students, I worked on this for many years, try to understand how the genetic network is induced after you do DNA damage and which proteins and genes are involved. Now, I come to the Niels Bohr Institute. We have cherished Niels Bohr a lot, famous physicist. He said, simplify, and we simplify these networks. Physicists like that, biologists don't always like that. Very interesting. I've talked to this many conferences. Biologists say, why do you want to simplify? You have much more knowledge here. But I say, we cannot. Uri wrote one equation. Here you have 25 equations. We simplify to 3 equations. I come later. But it's an interesting story. We can discuss that. So here we have the experiment you damage, and then they form these condensates or foci with a lot of repair proteins around the damaged site. And our theory goes on how the oscillations of p53 is related to repairing of this, and it was published in Cell 1.5 years ago. Now, as I said, p53 regulates around at least 200 genes. You can have also sustained p53 level, and that can lead to senescence and apoptosis. It's well known. And if you damage, you have the oscillations, and then you have the DNA repair. But of course, if in order to prevent aging, it should be so that the DNA, if it's not repaired, it should kill the cell, it should go apoptotic. And that happens luckily in 99% of the cases. If it doesn't, it induces cancer. now I think it works here. Yeah, here I show the oscillations from Galitla Harvard Medical. The period is around 5.5 hours, and the cell can also, here's one day, it can oscillate for almost a week. And you see the inhibitor MDM2, p53 is a transcription factor. It promotes MDM2, but it inhibits at the same time. And you see it oscillates a little bit out of phase, as it should. Now here comes our model of the simplified, as I say, Niels Bohr model of p53. Looks very much like Uri's of p53. It has a production, it has a degradation that saturates, as we just learned, and then it binds as a dimer. It produces MDM2 that translate and come back up here and work negatively. So, of course, this is a beautiful model in ordinary differential equation. Some physicists know, biologists might not know. Anyhow, it's quite, in principle, simple, and we can, my students here, look at the oscillations. You can have oscillations of different amplitude, of different periods, and of course, by adjusting all the parameters, we can explain the experimental data really, really well. So, however, now the experimentalists at Harvard come and radiate with gamma or X-ray. Gamma makes more damage. How much damage you do in a cell? They typically do between 20 and 50 damage sites. And then we see that repair proteins will be recruited to these sites. And there are lots of recent evidence, and I just run them here. You see rapid accumulation of p53, paper 2 years ago. And over here you see rapid accumulation at damage sites at 53BP1, a very famous repair protein. You see here. So there are lots of evidence that these condensates of foci will form. By the way, there's lots of evidence that telomeres also form these condensates of foci, and of course extremely related to aging, which we're just getting into. Now, as I said, we have these droplets or condensates around damage sites. They have a certain size. It turns out if the size of the droplet is larger than a critical radius, it will grow. It will size of less than a critical ratio, it tends to shrink. In physics called Oswald ripening, an old theory by two famous Russian physicists already back in the 60s. And this sometimes, I show here simulation, does it work? Sometimes I have to do this. I think it works. Here's my student, Alexander Luchetti, have done this simulation. You see here that there are damage sites, and here this droplet will tend to grow and outcompete the others. There will be lots of repair proteins in these droplets, but they will give it to that guy, and that will take all the repair proteins. It's when we simulate these equations directly, and you can ask me later, I don't have time to get into it. There's a diffusion, there's a density inside the condensate and outside of the repair proteins, and so on and so forth. However, our whole idea is that, well, I here have another paper, recent paper, that actually people did see to a certain degree that these condensate after damage will grow and form a damage site. There might even be a scaling law, which some of Laszlo will talk about, and so on and so forth. So definitely people see a lot of evidence for these foci growing after damage. However, now we induce p53 oscillation coupling to this equation I showed before. There we assumed constant p53, but now p53 oscillate after damage. What happens then? Then you see, droplets form, but they shrink again. Now droplets form again, but they shrink again with a period of 5 hours as the oscillation induces. Now droplets form and repair around damage site, and they shrink again. Aha! Very nice. So here you see that the condensates form with repair proteins, repair the damage site, shrink again, form another pace, repair, shrink again. You don't see one droplet outcompete the others. Very nice. So here's a balance between the oscillations and the droplet forming, which will lead to the fact that the repair is optimal. This is the whole idea, is what I'm going to say. Maybe I say again. P53 oscillate in density. High P53 recruit a lot of repair proteins to the damaged site. They repair. Low P53 retract the repair proteins. And then P53 goes up again after 5 hours. Then they recruit repair proteins at the damaged site. It's repaired and going back. So due to this delicate balance between the oscillation and the repair, we will get optimal repair. And this is what I will show here. Here you see what I just showed before. And what am I? Maybe I speak too fast. What's the time, Max?
You're good. You have 9 minutes left.
Oh, boy. I was too fast. Then I go a little slower. So here you see, I told you before that you see the condensate form at our damaged site. They shrink, they form, they shrink, and so on and so forth. Then, of course, we did a model of the repair. And the repair, we assume there are some steps. We call it Markov steps in mathematics. And due to other repair proteins that are on the damaged side. And then, of course, we can measure in our model how much damage is left and how much is the repair. And what we found is a very interesting curve here, that here we have the period of the external oscillation of P53. It goes from actually almost a day down to around 1 hour. It's the window here. Up here we have, for very, very slow oscillation, we have very bad repair. For very fast oscillations of the P53, we have very bad repair. We found a very, very interesting window where we basically repaired all the DNA damage, actually in our model to 0. All damage is repaired in a window of the external oscillation going from around 1 day down to 1 hour. Aha! And since we know that that P53 oscillates with a period of 5.5 hours, we know that experimentally that sits exactly here. We have exactly that the experimental observed period leads to complete repair of the damage, and of course then prevents aging, exactly for this period that the experimental observed. So a good thing. Just tell the story. 2 years ago we sent this to Cell and actually got incredible good reviews back. So of course we're very happy, said that's a great idea. I have to say myself, I looked at, in particular when Uri and Galit first showed the oscillation, what does it mean? What does the oscillation do for the cell? And since they start to oscillate when you do damage, you might think that the oscillation have to do with repair, but we couldn't understand it before we found the idea that the oscillation in fact induces these droplet of repair proteins around damaged sites. But actually the very nice editor said that it's a fantastic idea, but it would be great if you have some experimental validation, of course. So here I just say that we had a theoretical hypothesis that the oscillations leads to repair. We have the idea that the droplets form around damaged sites and so on. Then we got a contact to a group in Taiwan by Sheng, who actually was 6 years at Harvard Medical School with Galit Lahav, and they did 6 different cell lines you see here. One is healthy, 5 are sick, for cancer cells. And they could actually, of course, induce oscillations by damage, but they could also induce sustained P53, more or less at least, by adding noddling. So, since our theory said that the oscillations are optimal for the repair, while constant P53 is not optimal for the repair, they could actually study exactly these 2 cases. And what you see here, there is an gamma X2AX level. They measure the damage. Some of you guys know much more what that means. But what you see that after they do the damage by adding NCS. You see for the oscillatory P53 after 24 hours, basically all the damage is gone. Whereas if you have sustained level all the damage is still staying there, and you see here beautiful droplets forming within the nucleus of the cell. So they actually confirmed our theory in a very nice way. You see here is just a PDF of the damage, number of damage sites. If you have sustained p53, you see you have a lot of damage sites left. Here the number of damage sites is around 20. By the way, the damage site, of course, as Uri said, are double-strand breaks. As we well know, single-strand breaks are easily repaired. The double-strand breaks are very hard to repair. And if you have oscillatory, you see that much fewer sites are damaged after 1 day. Okay, so that's the idea that oscillations of p53 recruit a lot of repair proteins to the damage sites. The oscillation will help that the droplets and the condensate around damage sites, they grow, they shrink, they grow, they shrink, except for the fact one will tend to outcompete all the others. I end by just showing, if I can. Why is that? Here it is. Just end that we have now a fantastic, another fantastic collaboration just published now in Cell Systems with Galit Lahaub Lab, and I just want to tell you the following, that we know that if you do DNA damage, then p53 oscillate. But then, some years ago, I got the idea and talked to Galit about it, that you could also oscillate try to oscillate a protein or whatever from outside that could couple to the inside oscillating, and then you could have two coupled oscillators like two metronomes on a piano. And the incredible thing is that that took us almost 10 years to finish this, but it works. So they do damage and they oscillate Notlin from outside, and we can find all kind of synchronized states between the internal oscillation that the cell of p53 and the external oscillation. It's called Arnold tongues, somewhat technical term, but not very complicated, because they could do the external oscillation from 5 hours, 4 hours, 7 hours, 8 hours, 9 hours, 11 hours. What you see is first you have the p53 oscillations after damage, then you come from outside with an oscillator, it immediately coupled to the internal oscillation of p53. And you see the different periods it coupled to the external one. Amazing. It's amazing for me because working in United States, University of Chicago, in fluids, we did that in 80s. And for me, it's amazing that we can do the same for cells that we did for fluids many years ago. They could also increase, and this is really Alba Jimenez who does this experiment, it could increase the amplitude of the external oscillator. Then you tend to couple more the two oscillators. And here you see you increase amplitude 0.25 micromolar, 0.5, 1, and 2. And that led to the fact that we could actually do what's called Arnold tongues. And these are the really different synchronized states you can have, that they have exactly the same period, internal, external, but you can also have that the internal has a period and the external has the double or the half. And that's both called 1/2 Arnold tongue and 2/1 Arnold tongue, and then we can synchronize from outside the internal oscillators. And here is, of course, okay, I'm not sure I can backdate. We both, of course, have a model of this, and we have experiment. And actually, our very good friends at Harvard Medical School asked, do you think you can see chaos? Maybe some of you don't know what chaos is. Chaos is, Uri talked about stochasticity, which, of course, we do exactly the same way as Uri told us. But you can have a deterministic system that also has stochasticity. It's called chaos. That's something very new in physics, new 30 years ago. And indeed, how do you do that? You try to find two states that are more or less the same. Do I have that on the next slide? Can I go back just a second? You try to find two states that are more or less similar from the beginning, and then they should diverge as time goes. And here you see both model and experiment, and we actually found an indication of that. There's a measurement of that technical term called Lyapunov exponent. I don't have to go into detail. So actually with this fantastic experiment, we can go from ordered state into chaos state and into, again, you can see something called period doubling, which we could see experimentally. And after, if you increase Noethlin amplitude from outside a lot, at the end you should come back to the 1-to-1, because that will dominate. That's the two metronomes doing exactly the same. So we had this fantastic agreement between theory and experiment. So I just will want to end that I have had a lot of collaborators, in particular my late mentor, Leo Kadanoff, University of Chicago, who 10 years ago asked me, why don't you do the same for cells as we did for fluids? Try to couple two oscillators. Actually, I also worked with Uri some years ago, and here are some papers, and here all our funding. So the thing what it has all this to do with aging? Of course, we know that if you have double-strand breaks and they're not repaired, it will induce aging quite fast. And it's very, very bad because it also induces cancer. And we have seen here that we believe that p53 is called is of course extremely important in cancer research and cancer theory. And we believe that the famous oscillations of p53 after DNA damage is exactly a way to repair DNA double-strand breaks and thus prevent cancer. Thank you very much.
Thank you. Got time for 2-3 questions. Well, those 3 people there. Oh, well, Vera, quick one.
So have you tried to make the oscillations slower?
Yes, I think I tried to show that if in our model, if you make slower oscillations, you mean longer period, right?
No, just to change the amplitude.
The change amplitude. Yeah, amplitude or period? What do you mean? Slower means period, right?
Yeah, period.
Our model shows that within that window you still have optimal repair. Experimental is not so easy, but you can change period with temperature.
Based on the information you collected, did you have any insights into a possible hypothesis on why breaks are sometimes successfully repaired and why they are sometimes not successfully repaired? Whether it has to do with this mechanism of oscillation, if sometimes it's not at the right frequency?
Great question. Of course, sometimes it's not repaired. And in our model, relating to what you said, if the period is not changed, you don't get optimal repair. It could also, why? Because, for instance, if it oscillates too fast, then you don't have time to repair. The repair proteins don't have time. If you oscillate too slow, then, as I showed you, you will repair one place and not other places. It's a delicate balance. And of course, sometimes, as we know in biology, it just doesn't work, and that's not good. But we know from model a lot about your question.
One question here and then one here.
Hey, just a couple quick comments. One's actually related to Uri. I think the yeast data set he's referring to is either Nan Hao Lee or George Jansen's, but I don't think we have large data sets. I don't think it was ours. So I want to give credit for that. I think it's great data. Regarding this, I think that we've not valued the idea of dynamic ranges and aging enough in the longevity field. I think that most of the interventions that we've to date developed have been about maintaining dynamic ranges that are present in youth rather than turning pathways up or down. I think that would refer to TOR, would refer to autophagy.
Yes.
Or even coming back to yeast, it would refer to the normal metabolic oscillations. So I think this kind of thinking is really helpful.
Thank you.
Helping move the field.
You hit just in my stomach here because I work on dynamics in mathematics, right? And of course you can say, okay, a constant level is a constant level. What's the next step aside a constant level. The simplest thing, mathematically, is when it starts to oscillate, right? And lots of things in our body oscillate, and I think, thank you very much for this comment, that oscillations will be extremely important you talk about yeast and calcium oscillations, and all kind of oscillations are extremely important. Thank you.
Hi, very nice talk. So this might be like a question outside of the scope. But P53 modulates various other like stress processes, or it's implicated in autophagy, inflammation, cell cycle arrest, apoptosis. I'm wondering if it's known how these oscillations influence these other stress responses.
Yep. Okay. Absolutely. So, as I said in the beginning, if the damage is not repaired, then the cells should go apoptotic, or at least senescence. And they experimentally see a lot of cells going apoptotic, stop oscillating, going apoptotic. And that's, in my opinion, the healthy response, right? So if you don't manage to repair DNA, we don't want the cells to double. That causes cancer. And so we know a little bit about that.
Perfect. Thank you. Thank you very much for walking us through the oscillations.